Hello, everyone. Thank you for joining. We are just going to let the attendance list populate for a second, so please bear with us while everyone joins. And I'm very happy to be here with my co-hosts from the Connecticut College Arboretum, the Arnold Arboretum, the Morton Arboretum, and the Arboretum at Penn State. Very excited to be presenting this to you tonight. We've got 45 people logged in right now. So we can maybe just wait another minute, because I think we'll get a few more. All right, well, while that attendance list populates, we can get started. I'd like to thank everyone for joining us tonight. My name is Mike Beletsky. I'm the Director of Education for Bartlett Arboretum and Gardens in Stamford, Connecticut. We are very excited to have our two speakers here tonight to talk about conserving the American chestnut. The American chestnut was a staple of the American forests for many, many years. And in the early 1900s, chestnut blight basically wiped out the entire population. And we have two experts here tonight to talk to you about the chestnuts and about the work that they've done with them over the past several years. First, we'll have Dr. Sandy speaking. Dr. Sandy is from the Connecticut Agricultural Experiment Station, and she's been working there since 1966. She first worked in genetics, but now is in plant pathology and ecology. And she's done various work in all sorts of genetic studies of fungus and methods for detection of extracellular enzymes produced by fungi. So we're very excited to have her here introducing chestnut blight disease and the fungus that causes it. We also have Dr. Oakes here with us, and Dr. Oakes is a research scientist for the American Chestnut Research at the State University of Environmental Science and Forestry in Syracuse, New York. And that is a mouthful and a wonderful title. She is optimizing American chestnut elm agrobacterium that's mediated transformation protocols for introducing elm yellow resistance, thanks to a research grant that she has. So we're very excited to hear about the research that she is doing with the diseases that cause these types of problems. I'd like to give my co-hosts the opportunities to introduce themselves and their arboretum before we get started. So why don't we start off with Megan?
Thanks, Michael, and thank you to Dr. Sandy and Dr. Oakes for presenting tonight, and to all of my colleagues from other arboreta for collaborating on this webinar about this critical tree species with us. So I'm Megan Dunning. I'm the manager of adult learning programs at the Morton Arboretum, which is in Lisle, Illinois, just outside of Chicago. It was established in 1922. Today, the living collections of the Arboretum cover 1,700 acres with more than 200,000 live plants, representing nearly 4,300 taxa from all around the world. We hold those collections in order to inspire visitors to learn about, plant, and protect trees, and we champion the world's trees through plant collections, science and conservation, education and outreach, and we lead scientific research on tree health and tree improvement all over the world. So it is a real thrill to be part of this program tonight, and I'll tell you a little bit more about some of our other initiatives towards the end of the program.
Tim, do you want to go ahead?
Sure thing. So hi, everybody. Very happy to be here. Very happy to be collaborating with my colleagues at these other wonderful gardens. My name's Tim Gould. I'm the Public Programs Manager at the Arboretum at Penn State. If you're not sure where that is, Penn State is located right in the middle of Pennsylvania. We are a fairly young arboretum. We're only about 10 years old, but we are a 370 acre arboretum. We have about 10 acres of managed botanical gardens and then plenty of natural lands. So if you haven't been here before, I highly suggest that you come. It's kind of a hidden jewel here in Pennsylvania. And we are constantly growing, constantly under construction. So there's something new every time you come visit. So if you're from the area or within a driving distance, I invite you to come out and see us.
Thank you. Maggie, do you want to say anything about Connecticut College Arboretum?
Sure. Thank you, Mike, for organizing this program. My name is Maggie Redfern, and I am the Assistant Director at the Connecticut College Arboretum. We are a small liberal arts college on the southeastern coast of Connecticut, and we have a 750 acre arboretum that includes our campus, several hundred acres of natural areas, and our native plant collection, which is the original arboretum, or our original arboretum, I should say, that was established in 1931. And since 1931, just 90 years last Friday, we celebrated our 90th anniversary. The Arboretum has grown plants native to Connecticut and the eastern deciduous forest. Our wildflowers are looking great right now, and we're looking forward to mountain laurels and many of the other native trees that will be flowering and providing beauty for us all summer. I also want to wish everyone a happy Public Gardens Week, because I don't think anyone has mentioned that yet. But part of the impetus for us all getting together was to celebrate public gardens where we work, where we live, where we enjoy spending our time. And it's great that so many of you are joining us here today to learn more about chestnuts. And one side note for Dr. Sandy, we've been happy to have her join us at the Connecticut College Arboretum in the past to help us with our butternuts and our chestnuts. And I'm really looking forward to hearing both of our presenters speak this evening. So thank you, everyone.
Thank you. And I just want to quickly mention our fifth co-host that is not able to join us this evening, the Arnold Arboretum of Harvard University. We're very happy to have them as our co-host as well. And I'd just like to mention one or two quick things about the Bartlett. We are a 93 acre nature sanctuary in Stamford, Connecticut. We have a number of notable trees on our property, including several Connecticut champions. And one of our notable trees is a chestnut that was actually cultivated, or I should say, grown by Dr. Francis Bartlett himself and named for him, and it's called the Bartlett chestnut. And we have several of those offspring chestnuts, and they are not an American chestnut. They're a Chinese hybrid variety, but they are still quite fascinating. Oh, and Sandra's shaking her head, so maybe she knows something that I don't know.
They're pure Chinese.
I'm sorry?
They're pure Chinese.
They're pure Chinese. So, well, without further ado, since you are the expert, I'd like to turn it over to you so that you can tell us as much as you'd like to about chestnuts.
Thank you. Okay. Michael, you have to let me share the screen.
You have the ability, try again.
Okay, I'll turn this off and try again.
And I just want to remind guests while we're waiting that if you have any questions throughout the program, you can ask them using the Q&A feature. Or if you'd like to communicate with us, you can use the chat feature and we'd be happy to communicate with you that way as well.
No, you must not have clicked it. Let me try one more time.
Oh, there we go.
There we go. Okay. Now I am sharing the screen. Nope.
We have your Zoom, we have your web browser open currently.
I know. Okay. Let me stop that and try again. This worked before.
We all have these technology challenges.
All right. You want me to hit share? And I'll shrink that, and there it is.
Wonderful.
Okay, we're on.
Perfect.
This is a real pleasure to be able to talk to all of you and to join with Allison to talk about some interesting new work that's going on with chestnut, and which I think everybody needs to hear more about. So it's a good thing to give you a little bit of grounding about chestnuts and the blight. American chestnut trees were native to a fair amount of the eastern United States. I liked this graph that came from Bison and Foster because it shows you in green the area that a man named Little reported as the native range of American chestnut trees. It's interesting that when Lucy Braun did her studies of the forests of the eastern United States, she was a little more specific about where chestnut trees were found. And the areas that are spots on this map are places that Lucy actually surveyed very carefully. Any place where there are dark spots, she recorded American chestnuts as being absent. Where there are pink spots, they were present, not abundant, but present. So you can see that American chestnuts were present in her study through most of the range that Little had designated. And then in the mountains of the Appalachian Mountain Range, she found places where American chestnuts had actually been dominant. And this tells you that the presence of American chestnut, though they were present in a lot of the eastern United States, they were very important to the people in the Appalachian Mountains because they were a dominant tree. They were a tall, very tall, straight tree, and so they were very important for timber. Now, the other thing that people tend to forget is that American chestnuts actually have very small nuts. So very early on in the United States, in the 1700s, people who were interested in chestnuts for the nuts started importing European chestnuts. So European chestnuts were planted and put in orchards all over the East Coast and in many other places in the United States. And I've included these pictures because I get samples all the time from people who say they found a chestnut tree and they assume it's an American. Usually it's actually either a European or a European hybrid. So I put this picture in to help people think about what American chestnuts look like. These are American chestnut leaves and twigs. And I think the characteristic that is most useful is early in the spring. These little leaf-like structures are stipules. And you can see that on the American chestnut, the stipules mostly point backwards away from the tip. This is a European chestnut over here. And I'm sorry that the picture's not better, but these stipules, though they're also kind of skinny, are facing towards the tip of the branch. And that's much more characteristic for European. So that's my handy way of telling them apart. But people need to realize that European chestnuts were planted all over the eastern United States, and there were hybrids of American and European that made excellent nuts and were planted in orchards everywhere. So both American chestnuts for their timber and the Europeans and their hybrids were fairly common in the early days of the United States. By 1900, people all over the East Coast certainly knew about them. But then when Japan was opened up to exploration, there were many plants sent from Japan to the United States. And as often happens, human beings just don't think about the problems they're introducing. And these Japanese chestnut trees brought with them a disease which certainly infects the trees, the Japanese trees, but doesn't cause them serious damage. This picture is a Japanese chestnut that was planted in Old Lyme, Connecticut. The first actual importation came into New York, and the owner of the nursery gave the seeds to his coworker who planted them in southern Connecticut. And because the trees came up quickly, they were small, they produced large nuts very early on. Everybody was very excited about Japanese chestnuts because they were different. The nuts were very large. And this 1876 tree was only one of many that were distributed by this particular nurseryman. I found several throughout Connecticut. And the interesting thing about Japanese chestnuts is that, yes, they do get chestnut blight. And this particular tree has cankers all over it, disease caused by the chestnut blight fungus, which came in with this tree. But they're simply roughened areas on the bark, and the tree is doing fine. So what about this disease? Because when it got into places where there were American chestnuts or European chestnuts, both are very susceptible to the disease. The fungus gets into broken branches or wounds on the tree, and any place where there's a wound, the fungus gains an interest. It can grow first on the dead tissue, and then it starts growing in and underneath the bark until it has grown all the way around the stem or the trunk. It pokes through the bark and produces these little orange cushions of tissue, and that's where the spores are formed. The other thing that's happening here is that as the fungus is growing under the bark, it's killing the cambium. Now, chestnuts are ring porous trees, and that's important because in ring porous trees, the vessels that are containing, transmitting water up from the roots and nutrients down from the top, those vessels in ring porous trees get blocked up at the end of the year. So in the spring, when trees are starting to wake up again, they need those vessels to provide water up to the top. If the cambium is killed, no new vessels can be made. Normally, the cambium would be ramping up in the spring and producing very large vessels to transport water up to the top of the tree. But if the cambium is killed, no new vessels are formed and the tree dies very quickly. That's a really sad fact of chestnut blight disease, is that once it gains a hold in an American or a European chestnut, the trees are killed very quickly. And they're killed back to the base, but not to the root collar. So the root collar, which has hundreds of dormant embryos, produces lots of sprouts, and those sprouts will start to grow for a while and become wounded. And because the chestnut blight fungus is now with us and apparently will always be with us, the trees become infected, the tops die, and they sprout again. And this continual dying and sprouting has certainly captured the imagination of scientists for many years. And people started trying to find a cure. Obviously, the first thing that they thought of was planting lots of Asian chestnut trees, because both Japanese and Chinese chestnuts have quite a bit of resistance to the blight. So lots of Asian chestnuts were planted. But because people were continually importing these other trees, the blight very easily spread. Yes, it did spread to some extent on creatures carrying it around, but I suspect that most of the spread was because of people planting trees that were infected, and it then spread out into the forests around the orchards where the trees were planted. The fungus was first identified on Long Island here in New York City, and by 1908, there were diseased trees found everywhere there are red spots. At that point, people started looking for it, and by 1911, it had been found further out. By 1913, more surveys were made and more infections were found. By 1930, it was pretty well through most of the edge, at least, of the range of chestnuts as defined by Little. I think this is one of the best graphs I've ever seen of how chestnut blight actually spreads within a planting. This is a place in Pennsylvania. The stripes that are sideways indicate a forest area. This is not an orchard. This is a forest with maple, oak, and chestnut. There was one tree found with blight in 1908. No way of telling how it got into the middle of this forest. It could have come on the feathers of a bird flying in, on the feet of a small mammal, something walking up and down the tree. By 1909, the following year, there were 3 trees with chestnut blight. By 1910, 14, and then as you see, by 1912, 166 trees in this forested planting were infected with chestnut blight. So you can see that once the fungus got into a forest planting, it spread very quickly, probably on all of the creatures walking up and down the trees, carrying the spores to other trees where there were wounds for the fungus to start growing. The next thing that people attempted, of course, after they planted all of these Asian chestnuts, they realized that the Asian chestnuts could not possibly compete in the forest because they had been primarily bred as orchard trees and they were short and spreading. And the attempts were begun to make crosses between Asian chestnuts with a fair amount of resistance and American and European chestnuts to get resistant trees that might be more like timber trees. The USDA had a breeding program that unfortunately was stopped about in the 1950s. The Connecticut Experiment Station started breeding both timber and orchard trees for resistance to blight in 1930. And the program has continued. Even though I'm retired, I'm still working and I'm still making crosses and working on finding some way of using breeding for developing a timber type chestnut tree, though a lot of the Connecticut effort and mine as well has gone into breeding orchard trees. But breeding for timber trees is a long-term project. We heard in the middle 60s about a phenomenon in Europe where chestnut trees were recovering. So after reading several papers by Europeans, especially a scientist named Grant in France, we decided that we really needed to look at this curing phenomenon that they had found. And Grant reported that the cure was because of something wrong with the fungus. It was the same fungus, the chestnut blight fungus, but it had something wrong with it. And when he put pieces of the diseased fungus into killing cankers, the cankers stopped expanding and the cankers seemed to be cured of the disease. What happens here, you can see are very swollen areas on this tree, these trees. These are places where the curing strains, the non-virulent strains, were put around these cankers, and the cankers that otherwise would have killed the tree the tree has responded by producing a vast amount of callus tissue and walled out the fungus, so the tree's able to keep living. I imported some of these curing strains in 1972, and we very quickly found out that the thing that was wrong with the fungus was actually a virus. And we also showed that the virus could be transmitted from one strain to another. And that's how the virus is able to be a curing phenomenon and keep trees alive. We have trees that are in a small orchard, American chestnut trees. They were planted in 1976. And by then we had permission to use these curing strains, these non-virulent strains outdoors. So we tested them in this small orchard. We treated every canker we could reach for 4 years in a row. By 1981, we had put the non-virulent strains, the hypovirulent strains, into many cankers on these 70 trees in this small planting, and those trees are still alive and well. Now they're in an orchard setting, they get plenty of water, they're not under any competitive stress, and that was great. But when we started using this in the forest, I was so happy. I was sure we were going to bring back the American chestnut. But what we found was that the strains, though they could produce these large swollen cankers on the forest trees, the trees had so much competition that they weren't able to produce enough callus tissue to wall off the fungus. And they ultimately died. They sprouted from the base, just like chestnuts always do. And so the cycle goes on. It was clear that the hypovirulence, though it was useful in an orchard, was not going to bring back American chestnuts as timber trees in the forest. Then in 1978, at a meeting, a graduate student, David McCarroll, and his advisor, Ivan Thor, reported that they had found that the blight fungus produced oxalic acid. They suggested that this might be important in the way the fungus grew in the trees and was able to cause cankers. And then Evelyn Haber and I at the experiment station started doing tests on both the killing strains of the fungus and the non-lethal strains, the virus-containing strains. And we found that the killing strains produced a great deal of oxalic acid, and the virus-containing strains produced very little. Now, oxalic acid has the ability to chelate the calcium out of the bonds holding the tree cells together in the bark. Besides that, because it's a strong acid, it's killing the cells that have been bound together. So the fungus is able to use the nutrients in those cells and grow between the cells and continue to grow all through the bark and kill the cambium and a lot of the external tissue. And we felt very strongly that this was evidence that the oxalic acid produced by the virulent strains was extremely important in the ability of the fungus to kill trees. So what's the next logical step? Yes, I'll keep on with the breeding, but obviously, if oxalic acid is so important, there ought to be a way to find a gene that we could give to the trees that would allow the trees to break down the oxalic acid that the fungus is producing. So over to you, Allison.
Okay. I'm gonna thank you for the wonderful introduction to chestnut trees and the blight. And let me see if I can swap this up real quick.
Yep, it changed over.
Okay. And then, okay. So yep, thank you again, Dr. Sandy. And I'm Dr. Oakes, sort of popping in here. I've been working with the American Chestnut Research and Restoration Project at SUNY ESF since 2006, when I needed a job and started washing glassware in this really neat lab that was doing some really cool work. And so I'm gonna pick up from where Dr. Sandy left off, and just a little bit about how it was kind of a serendipity that she and her team found the significance of oxalic acid, just as the New York State chapter of the American Chestnut Foundation was coming to Dr. Bill Powell and Dr. Chuck Maynard at SUNY ESF and saying, hey, is there a genetic engineering approach that we could use? We hear all about it. There's a bunch of companies doing this commercially. What about genetic engineering for a conservation purpose? And so things lined up in that there was a target, the American chestnut, and this oxalic acid the fungus is using to break down the tree's defenses. And what about, like Sandy said, what if there's a gene that could produce an enzyme that would snip that oxalic acid into? And so what Bill and Chuck found, sorry, Dr. Powell and Dr. Maynard found, too familiar, was that there is an enzyme. It's called oxalate oxidase. So oxalate is oxalic acid, and then the oxidase is the enzyme clipping part. And they found that there was this very well-studied enzyme in wheat, not gluten, totally different family group. And what it does is it takes the oxalic acid and breaks it apart into its components. And the great thing about this enzyme is that it is not a pesticide. It doesn't hurt the fungus, destroy the fungus, kill the fungus. It's not a fungicide. Okay. So that really reduces the chance of the fungus developing resistance to it, because there's not a heavy selection pressure on this particular mode of action. And so what this ends up doing is that instead of killing the fungus or removing it outright, it just changes the lifestyle of the fungus. The fungus still infects a little wound on a tree and goes through its life cycle, but it can't spread, or at least the idea was it couldn't spread if the tree is actively getting rid of the fungus's main weapon. And so we've looked a lot more at this enzyme, oxalate oxidase, and many plants have this. It's a very common defense mechanism against fungus. So not only in wheat, but it's also in corn, barley, and rice. So all of our main grains contain oxalate oxidase. Peanuts, strawberries, walnuts, tomatoes, cacao, sorghum, potato, apricot, dates, oil palm beef, so many different plants, basically all monocots like grasses, have this. We've seen it in, we found similar genes in native North American grasses and also in black walnut. So it's variations on a theme, but many plants have this type of enzyme in order to protect them from fungus that use oxalic acid as a weapon. And that's all we know so far. I'm sure there's many more to be discovered. But so the idea is we have this gene that would then produce the enzyme, but how do we get that gene into a plant? Well, it's super easy. You just, genetic engineering, scientists do it all the time. It's very quick. It's I kid, it's extremely long process, and many people doing many different parts of the process and optimizing them all take years. So, well, to start, you choose your gene of interest, which we have, the oxalate oxidase, and you prepare it in this little cell called agrobacterium. Now, Agrobacterium tumefaciens is in the wild, lives in the soil, and is a natural plant genetic engineer. What this bacteria does is inserts a little bit of its own DNA into a plant, and usually that creates crown gall. But what scientists realized is, well, what if we could take out those crown gall causing genes and use Agrobacterium to deliver the gene we want instead? So Agrobacterium figured it out itself, but scientists have gone in and use it now as a tool to help them insert the gene they want into a particular plant. Okay. On the other side, we need to get some American chestnut tissue that we can insert into, and what we found that works for chestnut so far is immature embryos. So we go out to our trees, like a wild type chestnut that we think is really neat. There's a very tiny window in the summer, and you go out and collect these immature little incredibly prickly burrs, cut them open, pry them open, and you can see this little, it's not even brown yet, immature chestnut. You take that and you sterilize them. And so you get them, try and wash them with bleach and detergents and get them really clean. And then you're working with them in a sterile environment. We're gonna cut open that tiny little nut and try and find some teensy little embryos. So we can see the one has already started to become dominant here, but ideally, so a little earlier than this, you'd pull these out and put them on a growth medium that would allow the embryos to continue growing. And this is a very difficult procedure, and it's like a 1 in 1,000 success rate. But eventually, if you throw enough grad students at something, you'll succeed. And so we get these little growing clumps of little tiny embryos that are all growing happily on a plate. And over here, we prepare an agrobacterium line that has the gene of interest that we want in the bacteria ready for it. It's just going to insert it like it would do in the wild. And then we smoosh them together, very, complicated scientific terms. Then we use a selection to kill off any tissue that does not contain our gene of interest. And finally, we get little clumps of chestnut embryos that contain the oxalate oxidase gene. We call it OXO for short. So hopefully that won't be too confusing. We have our chestnuts with OXO, our fungus fighting enzyme. And then we are here in the program. Then we would take these embryos and we confirm them via molecular biology techniques over in the lab. We got to check, make sure it's there. And then when we pass that, we go back to tissue culture. Okay. Once we know those embryos do have the gene of interest, then we take them and we convince them to become something different. We induce shoot formation off of those embryos. So by micropropagation, we can use different media, so different combinations. This jello stuff in the bottom here, we can manipulate. This was my PhD. I love this. We can manipulate the sugar or the hormones or the vitamins or even the salt content and convince plants to do different things and grow in different ways just by what they're surrounded by. And eventually, if you do it right, you get a little tiny green shoot coming up, and then it starts growing, and now you have a tiny little chestnut tree in a tiny little sterile box, and you can continue propagating and growing them on infinitum. You can make as many as you want. But we stop and we go and check and make sure that the gene is being expressed, that the gene is actually producing that oxalate oxidase protein. And then once we're sure it is, we start doing shoot production. We can multiply up the plants. A lot of arboretums actually use micropropagation to help conserve and propagate up very important plants that they're trying to save. And then we induce rooting, which in chestnuts was worth an entire PhD dissertation. Then we acclimate these things in growth chambers. So we gradually reduce the humidity and get the plants to start producing bigger and more mature leaves. And finally, when they get big enough, we move them into higher light growth chambers, fertilize them, and eventually they go to the greenhouse, and then we acclimate them a little more, and then they eventually go out to the field. So it's a long step for making genetically engineered trees for sure. Yeah. And then we check them again, and this time we do a leaf assay to make sure that we see blight resistance. And so it's similar to like a small stem assay that we do here. So we want to check and make sure before we plant stuff in the field, are we seeing some resistance? So over here we have an American chestnut that is, you can see there's a very big blight canker. The original wound was here, and the blight has spread up and around, and it's shrinking in here, and the tree is going to die above this point. Compared to a Chinese chestnut, the blight gets stopped, okay? The initial wound is here, but it sort of builds up a wall, you know, and the fungus can't get past. And so the Chinese, it looks ugly, but the Chinese chestnut's gonna survive this. On our very first American sort of chestnut here, where we inserted this gene, we saw, okay, it's a little bigger than a Chinese, but it's a lot smaller than the American, the pure American that has no oxalate oxidase enzyme added. So this was our first sign that, okay, we're onto something. So the next step was, well, let's try and get more events and see if maybe if the gene goes in a different area in the genome, more enzymes expressed. And what we found is over the successive generations, we found higher and higher expressing events. And then as we expressed more of the enzyme, the cankers got smaller. So this is the canker, this is the expression over here, and the canker size going down. So in our more later assays, we were seeing we were killing off all the American chestnut above the wound. All of our transgenic chestnuts with the oxo were surviving. And then even over here, the Chinese chestnut, some of them actually died back. They were actually killed by the blight. If it's a severe enough infection, the blight can kill the Asian species. But this was really, they're really short because we were standardizing via diameter, and Chinese chestnuts are a little thicker on the stem. So this was really promising. We're starting to see these big, beautiful chestnuts that look nice and chestnutty. And then we did a comparison after breeding these, which I'll get to in a second. We looked at siblings, okay? So two trees from the same parent. We wanted to know, does this gene get passed down from parent to child? The answer is yes. So I'm going to show you, this is the sibling that did not inherit the gene of interest, okay? And then this is the one that inherited the OXO gene. Okay, so it's a 50-50 chance. They might end up with it, they might not. And you can see the difference here between, here's a little canker, got walled off. The fungus, without that oxalic acid, the fungus can't go rampant through the tree. It can't kill the tissue. It just makes a little canker. We see a little of the spores here. It's gonna do its little life cycle, you know. But compared to this other same mother tree here, same dad, same pollen, but this one did not get the gene, and the level of damage is just, yeah, big comparison. Okay. So is it heritable? Can if we give these the trees the gene, and then it can get inherited by the offspring? And the answer is yes. So we have been looking. We're now on generation 3. We're in the great grandkids, and we're still seeing full resistance. If there's one copy of the gene present, then the tree is resistant to the chestnut blight. The enzyme is being produced, and the tree. So when I'm talking about the 50% here, is if I've got one tree and we inserted it one place on one chromosome, and then when that tree crosses, half of the kids get that chromosome that was inserted, has the OXO insert, and half the kids get the chromosome that did not have an insertion. So there's just one floating around. Eventually, we'd want to breed trees so that both chromosomes have the gene of interest, but we want to outcross as much as possible before that happens, because we don't want to like not have a lot of genetic diversity. We want to have as much genetic diversity as possible in these offspring. Okay. So how we're doing that is we're using a high light growth chamber to push the trees to pollinate, to send out their catkins early. Usually in the field, we'd have to wait 5 to 7 years, but with the high light growth chambers, we can push them in 1 year and get our generation cycle going very, very quickly. And so these are the male flowers of the American chestnut. They look like, they say, like a big firework. And when the hillsides were covered with these, it would look like snow in July, just the amount of catkins all over. And so what we do is find our so this is rubbing some, it's as easy as rubbing it on a glass slide. We get a little bit of static, it's perfect. And then we go find the female flowers that look like these little, almost little pineapples here. And up here, the little, where we rub the pollen on and they get stuck on the stigma, and then they go down and fertilize the little ovules and all that good stuff, and we get chestnuts. So we're doing controlled crosses in our greenhouses and in the field in order to determine, like, so we know who the parents are, and we've got really massive pedigrees. Okay, so this is what our trees look like in the field, because that pollen is not cleared to be released. So if we apply pollen to a flower, we have to cover the flower in a bag and make sure none of the pollen and none of the nuts escape, because we are currently all under USDA APHIS permits, and we're not allowed to release any of the transgenic pollen or anything like that. So, and everything gets bagged up, weird looking fruit. Okay, and just as a timeline, like this whole process has taken, you know, decades. Okay, so you can't go from just throwing genes into a plant and throwing plants in a field willy-nilly. It takes a lot of time, a lot of effort, and it's been quite a long road to this point of showing that resistance is heritable and having all these generations occurring. Okay. So then in the fall, you go out, and here's Dr. Powell with his clippers, and we clip down the branches with the burrs on them, unwrap them, and count our harvest. Okay, so they're all the pollinations. We know who the mother and father is. It's recorded on the pollination number. Here's a branch where that burr, those nuts would have been gone. Squirrels love chestnuts. Rodents, deer, birds love them. So it's a good thing we put the bags on, otherwise we'd lose them all to wildlife. Okay, and then we have our harvest. We shuck them. We get rid of any of these. These are little ones that didn't take, like little immature unfertilized nuts. And then here's a gorgeous sort of triad. The American chestnuts usually occur in a group of 3 like this. And then here's a sort of what a handful of them look like. You can imagine roasting chestnuts on an open fire with these. And then we're going to put them in little peat bags, put them in the fridge, let them just stratify over the winter, and then we're ready to go the next spring. We plant out all of our little seedling chestnuts tagged individually with a unique number, and some lovely people get to run that spreadsheet. I don't envy them. And then we have our crop to plant out in the field the following year. And planting out is always a big communal activity. Everybody comes and joins in. We bring our kids, and it's really fun to see people of all ages really getting into the chestnut project. And then they take off like a shot. They grow very rapidly. I think our best coppicing was 7-foot growth in a year, and they're lovely, beautiful trees. So the idea that we have spawned with between the project and Alan Nichols and the American Chestnut Foundation at large is that what we want to do, we don't want to cross these, or we don't want to clone them. We don't want to have like plantations of all the same tree. We want to capture all the remaining genetic diversity that's out there in the population. So the idea is to take the pollen from the transgenic chestnuts and go pollinate regionally adapted trees, ones that have survived or just, you know, they have good genetics, and pollinate those, all different ones, like pollinate the Maine ones, the Virginia, the Georgia ones, and then collect the seeds from those trees, and then you're going to get different compositions of the genotypes. You're going to try and get all the different genetic diversity that's inherent in those local populations. And then half the offspring are going to be blight resistant, half not. It's going to be very easy to call them out. and then continue crossing out with different populations so that we don't lose the genetic diversity in the population. Because the last thing we want is like an inbred tree or a clonally propagated tree, because then the next disease that comes along is going to be a huge issue. Okay. So the other big question people have with these trees is that whether there are any off-target effects, whether or not the trees are going to affect other species. What effect is that having that gene in the tree, what is that going to do in terms of anything except blight resistance? So we have looked at whether or not, can mycorrhizae still form symbiosis on transgenic chestnut roots, or would the oxalic acid being broken down be a problem? And so far, there was not any difference seen between those. I'd show figures, but this gets very long if I start going into every figure from every paper we've done on all these. But the answer being no, we did not see any significant effects on mycorrhizal symbiosis. We did see less mycorrhizal symbiosis forming on Chinese chestnut roots, which makes sense. That's a tree from another continent and a totally different ecosystem, and so that would make sense. How about wood frog tadpoles? So there's gonna be chestnut leaves falling into vernal pools and falling into streams, and would this somehow affect tadpoles eating genetically engineered chestnut leaves? And basically found that nope, they actually did the best on the wild type American chestnut and the transgenic American chestnut, which are right about the same. They did worse on the hybrid chestnut and the Chinese chestnut. Again, that's a tree from a totally different continent that you're bringing over, and it doesn't have wood frogs, co-evolved with chestnut for millions of years, American chestnut, so it would make sense they do best on those. And we looked at things like growth and length and gossamer stage, development over time, and same thing that there was no difference between a wild American chestnut and a transgenic chestnut with the oxidase gene. Another study we did, what about bees? Are bees going to be affected by eating chestnut pollen? Turns out lots of different insects visit these chestnut catkins and eat chestnut pollen. And so with the help of beekeepers and a professor from Cornell, we went and gathered up we kind of knocked the bee pockets off the bees as they're returning to a hive. Not all of them, we're not that mean, but like a sample, we're able to collect chestnut pollen from our orchards. And then we had a bunch of little tiny micro colonies set up, each with about 5 bees in them. And apparently once you get to 5 bees, they act the way bees start. They start to build egg cells. They start to lay eggs and raise larvae and such. So here are tiny little bee friends. And we added a little bit of the oxalate oxidase enzyme to their food. And was there any other survival difference or any, did they have less offspring? And the answer was no. The presence of oxalate oxidase in their food did not, it didn't affect them really much. They still built egg cells and they still laid eggs, and they had the same amount of pollen and nectar use and things. The bigger difference was which hive we actually got the bees from made the most difference. Okay. So we've done, beyond this, a whole lot. We've done like a native plant growing around the base of the chestnuts. We've done native plant seed germination with chestnut leaves mixed in the soil. We're looking at physiology and photosynthesis and respiration, but so far we have not found any off-target effects. The transgenic chestnuts appear in all respects, other than blight resistance, to be functionally the same as a wild type American chestnut. And so it's just been really neat seeing these guys. This is a transgenic one that's maybe about 4 or 5 years old, and they just grow beautifully. Before we've had to cut off, we still do have to cut off all the flowers on the transgenic ones, and when they get too tall, we unfortunately have to cut them back to the ground. So one of the criticisms I hear is, well, you're not doing this on old trees. I'm like, well, we can't do it on old trees because we have to cut them down whenever they get old enough to flower. Hence why we're applying for the permits through USDA and EPA to allow them to get bigger so we can test what happens when they get older. So, and we have volunteers and students all throughout the year who come and work with us, and it's really just awesome working with this very talented group of people. So that we're currently under regulation by the USDA APHIS because we use agrobacterium, and that which is considered a plant pest, even though it's been changed to deliver the gene we want. They're worried about whether or not genetically engineered chestnuts would be a plant pest risk. Somehow, could we make an invasive tree. The FDA is worried about whether American chestnuts, if they're going to be eaten, are functionally the same as chestnuts for eating that are already on the market. And the EPA wants to know, is oxalate oxidase a pesticide or not? So those are the three agencies we're currently working with. We've submitted to the USDA APHIS. We've already had our open comment period, and we're applying to be allowed to grow the trees like normal trees, to not be under permitting conditions, that people could buy them, people could plant them in their yards and such. So all three of these agencies are working together on this, and they all have to kind of approve about the same time. But we're working with USDA first, and then FDA, and then EPA following. So the great thing about this project is how it's tied to conservation. And it's not, nobody is making any money off this. We don't have a patent. I think the only thing we're going to do is trademark, so that if people buy a chestnut, that they know that the chestnut was from us, but that doesn't generate revenue. So it's really, as soon as people get these trees and put them in their woods, they can do whatever they want with them, right? So the idea being that people are going to use this to restore the species throughout its current range, either through public plantings or private plantings or forestry initiatives. So other forest trees are also having a lot of problems, like American elm, for example, green ash with the emerald ash borer, thousand canker disease in walnut, bark beetle in the western pines. I mean, one of the reasons that lumber prices are through the roof right now is because of how badly the western bark beetles have decimated the pines up in British Columbia. Okay, and so all this research in progress, if you're using a genetic engineering solution to try and help a tree fight disease off, everybody's kind of staring at the American Chestnut Project going, what's going to happen? Is this going to be a viable tool for mitigating forest health problems? Okay. And not even just trees, but other groups we've been to coral conferences, because people are thinking about, well, we might have to genetically engineer coral to survive in warmer oceans, and other species that are currently under threat. Okay, so for native tree restoration for us, we're trying to branch out from just chestnut, and I'm currently working on an American elm transformation project, and I'm testing out a couple of possible disease resistance genes for elm yellow's phytoplasma, which American elm, even the ones that are Dutch elm disease resistant, are still completely susceptible to this elm yellows. And then another, we just got a grant actually to start working on Ozark chinkapin. So hopefully another relative of the chestnut that lives in the kind of Ozark Mountains, but it also was affected by chestnut blight. It got hit about the 1950s, 1960s, and was in decline. And so that's going to be our next sort of target trees to see if we can increase their disease resistance. Okay. So we've had a lot of great funding over the years. Very thankful to all our donors and collaborators. And I would love to take your questions. And then if you have questions for the project, you want to email, go right ahead. And then you can also, my own email's there if you want to send me something too. So let me see, is somebody gonna?
Well, yes, thank you so much. Tim, did you want to take it away with the Q&A?
Yeah, absolutely. So our first question here is, Sandy, I believe it was you that mentioned that chestnuts are rainforest trees. So our first question is this referring to just the Asian chestnuts, or does this describe other chestnut varieties as well?
Everything in the genus Castanea is rainforest. And that's the same as the oaks. There are lots of rainforest species. Maples, for instance, are diffuse forest. Their pores are not blocked up at the end of the year. And that's why you can kill an oak or a chestnut by girdling it, by cutting around and killing the cambium. But you can't kill a maple very easily like that. You can girdle a maple tree and kill the cambium, but because it doesn't need to make new vessels in the spring, it doesn't know it's dead yet. It takes several years to kill a maple tree by girdling it and killing the cambium, because it is not rainforest.
All right, thank you. So I'm going to do my best to attach names to these questions. I'll apologize in advance if I mispronounce your name. I'm going to do my best. So the next question here is from Ruda, and it says, for how long can Cryphonectria parasitica survive without the presence of chestnut trees?
Cryphonectria parasitica does very well on leaf litter in the forest. It can also infect other trees. It infects a number of oak trees. On scarlet oak, for instance, it causes a swollen canker-like structure at the base. But because it grows very well on dead wood, it can survive for a long time. And because every place where the fungus is already present, there are going to be chestnut sprouts because they keep sprouting from the base. And as soon as one of those sprouts gets wounded, the fungus is around. The spores will be carried there by creatures. Carpenter ants are very good at transporting chestnut blight, by the way. We did a big study on carpenter ants in my lab once, and carpenter ants will feed on the spores. And then if they meet another carpenter ant who asks for food, they regurgitate the contents of their crop, and there are live spores in that crop. So that's one of the ways that the blight fungus could be growing any place, producing spores, and can be transferred around by creatures.
All righty. So our next question here is from Laurie, and Laurie asks, can you please explain how you get many embryos from one nut?
Well, we're actually not trying to get the mature embryos. We try and get what we call pre-embryogenic masses. And so to get those, we have to get the very early on in development. And so there's actually a bunch of fertilized ovules in one nut, but only one will take over. And usually that's why when you plant a chestnut, you'll get one sprout coming up, but occasionally you get 2 sprouts. And we actually had 3 triples this year for whatever reason, but generally the rest of those like proto embryos will kind of fade away and one will take over and become the dominant. So.
Great. A question here from Vanessa, also for Dr. Oakes. How many years do you think it will be until we have approval to freely plant transgenic chestnuts?
You know, for 2 to 5 years, we've been saying 2 to 5 years. So it's looking good. The regulators are fairly positive. We've been in bureaucratic, like, you know, what's a term where you're just sort of stuck in an airport waiting, waiting room, bureaucratic waiting room for, you know, a while. I think we submitted our report back in October, and we were supposed to have an answer within 6 months. It's been 6 months. We're still waiting. So we'll see. I'm going to say 2 to 5 years because that's the, you know, boss approved timeline. Soon. If you want to know as soon as they are released, I highly recommend you become a member of the New York State chapter of the American Chestnut Foundation. So if you go to the American Chestnut Foundation, become a member, and then also sign up to be in the New York State chapter, and then you will get like first notice when all this stuff is coming around.
I think all of us will echo that. Please sign up and become a member.
Alrighty. Next question here is from Manuel. Manuel asks if we understand the mechanism of natural resistance to C. parasitica in the Asian chestnut species.
No, I don't think we do. It's pretty clear from all the breeding work that has been done that there are probably lots of different genes in both Chinese and Japanese chestnut that are responsible for resistance. And it's because of this large number of genes for resistance that the breeding work has been so frustrating. You can do a lot of breeding, and then you have to wait 5 years, 10 years to find out whether your tree is really going not only to be resistant to blight, but to grow well. So breeding any tree is a long-term project, which is why the experiment station started this in 1930, and here I am still making crosses.
They're definitely making progress on it because the genome of the Chinese chestnut has been sequenced and mapped, and we just actually published the sequence for the American chestnut, the genome as well, and so people are working on that to try and find what genes are involved in resistance. But like Dr. Sandy said, it's a lot of them, and trying to move all of those resistance genes from chestnut, from Chinese to American, is just not possible at this point. We don't have the technology to do like 25 gene switch. It's just so.
All right, our next question here is from Megan, and Megan asks, how would the FDA and APHIS definition of invasive species take into account the original territory that the American chestnut occupied and in some places dominated?
For them, they're worried about, so that was a big part of our reports that we're submitting, is the trees were here. But part of the worry is they like, well, now it's an established maple beech oak, forest. How is chestnut going to displace that? And it's like, well, that's kind of the point, because chestnut was there before. So it is a lot of interchanging, like, yes, this will become like hopefully a canopy tree. The invasiveness, they're worried about it more sort of moving, how fast is it moving out? So one of the things we're talking about is the transgenic chestnut going to move faster across the landscape than before? The answer is likely no, not before blight. No, we're not changed. There's, and that was part of our report, is whether or not this is going to change its rate of, exploration, colonization. And
Right.
Yeah. So did you do any like chestnut spread studies? How fast it moves?
No, but the general thought is that because the nuts generally fall only a short distance from the tree, that the spread within any forested area is quite small. Certainly blue jays will pick up chestnut seeds and carry them some distance and plant them. But even allowing for spread by blue jays, I think it is unlikely that chestnuts would spread out very quickly. Most of the, you have to remember, the human beings move things around. And I suspect that much of the presence of all of these different kinds of trees is due to human beings.
Great. All right. Our next question here comes from Joshua. Joshua says, awesome presentations. He said, Allison mentioned green ash. What about white ash?
I believe Purdue, their Hardwood Tree Restoration Center, they are working on both green and white ash at the moment.
Next question here is from Claire. Claire says, we know the location of a mother chestnut tree with saplings growing up around the site until they're brought down by the blight. Could we plant a transgenic chestnut in the middle of the site?
When they're approved, when we can, that would actually be a great way to start getting the gene into that particular gene pool. Yeah, that's ideally what we'd like to do, is if people can either, if they have their own trees that are big enough to pollinate, we can send them pollen, or we could send them seeds and they could grow them up themselves. And then they'd be able to exactly go to a place where there are chestnuts, plant one of the transgenic trees, and then the offspring from that tree are going to start being blight resistant.
All right, we've got another question here from Lori. Lori asks, how do you pollinate wild trees?
We don't. We have to pollinate trees that are within our plantings. And they are covered in blight, and it's a constant battle to try and keep the mother trees alive long enough to set seed. So they will actually, right before they die, kind of put all of their resources up into producing seeds. Yeah, so we can't go out and pollinate wild trees with the transgenic pollen yet, but that would hopefully be something we could do in the future.
All right, our next question is from Brian. Brian asks if the transgenic chestnuts decompose and return the same nutrients to the soil as the Native American chestnuts.
They do. We had a master's student actually worked on this, and we were in the same class a few years back writing papers on it. And she buried bags of chestnut leaf litter from both wild and transgenic and a couple other species of trees as well. And she measured the decomposition at different time points. I think it was like 1, 3, 6 months and then a year, and found that the transgenic chestnuts decomposed at the same rate. And they also, she measured the mineral composition, and it was the same between the two chestnuts. It was different between like chestnuts and maple and beech, but both the wild and transgenic chestnuts, there were no significant differences in decomposition.
We have an anonymous attendee who asks if you can please speak to the American chestnut's change in geographic distribution as it relates to humans or climate change.
Do you want to take this one, Sandy, or?
Well, because chestnuts are found all the way from Georgia to Maine, I think that climate change is not going to greatly affect the possibility of growing chestnuts. It may be too warm in Georgia in 30 years, but since chestnuts don't require a terribly long winter cooling in order to germinate, I don't think climate change is going to have a significant effect on where they can grow.
Mm-hmm. The one caveat to that would be Phytophthora cinnamomi is another chestnut pathogen, ink disease, chestnut ink disease, and that is killed by having the ground freeze. So if it gets warmer and that the ground doesn't freeze, that particular pathogen could start moving up from Georgia into other chestnut territory. And that's going to be a problem for a lot of different plants, actually. That's a very versatile pathogen. So
it certainly, Phytophthora certainly does already get to places further north. Mm-hmm. It comes in because people buy plants from further south, mm-hmm. and humans, again, moving things around. But fortunately, it does not survive the winters much north of New Jersey. Mm-hmm. But that would be a problem with the climate warming.
Yep, it's up in the mountains is okay too. Like higher elevations are not as prone to that. So, but chestnuts like the ridges and the peaks. Mm-hmm. So, yeah.
So Allison, you mentioned earlier that squirrels really like chestnuts. Ruta asks, how much do hybridized chestnut trees support native insects and other native species?
We haven't done a lot of testing on hybrids. We usually include a control in most of our experiments, usually an F1 control. So like a American cross with the Chinese, and we use that offspring to test. But we haven't done that particular experiment yet. So, Sandy, do you know about hybrids and pollination?
You mean the potential of moving the GMO gene into other hybrids? I think there would be no problem, because all of the chestnut species, there are at least 7, if not 8, species of chestnut there are four kinds, at least four chinkapins, which are in the same genus in the United States. And all of those species of Castanea will cross with each other. And I can't imagine there would be any problem of moving the OXO gene into any of the commercial hybrids.
And do you expect that those would continue to support native insects and other native species the same way that the American chestnut did before?
The hybrids behave differently. That is, we do see significant differences between the hybrid chestnuts and both the wild American and the transgenic American behave the same, but then the hybrids do occasionally go off and behave a little differently. Slightly lower mycorrhizal colonization, slightly lower tadpole growth.
But that's because of the genes from other species of Castanea.
Yeah, the European, the Japanese, the Chinese, because it's every time you cross, I mean, there's 40,000 genes in chestnut. And if I cross an American and a Chinese, I've now replaced 20,000 American genes with 20,000 Chinese genes. And those genes are optimized for a different environment, different pests, different pathogens, different soil, everything. And so even if you back cross the American a few times, you're still talking about genes moving around on the order of thousands. Whereas with genetic engineering, we added one for a very specific purpose. So the magnitude of change to the genome in traditional breeding is much higher.
Sense. Alrighty, we have one more question here from Laurie. She says that, you mentioned that transgenic trees cross with wild trees and the nuts that result have half the inherited genes and half don't have those inherited genes, and that it's easy to tell the difference. And she's wondering how we tell the difference. Do you just take the nuts out and test them, or is there another method?
The easiest method is to germinate them. When they grow up, they have a little leaf. We take a hole punch. We punch a little circular leaf slice, put it in a particular little chemical bath. And if the transgene is present, if the OXO is there, then it will stain dark around the edge. It'll be like a little dark ring around it. And if you take a wild type tree and do the same thing, it won't stain at all. So we have this nice little assay, which we hope to be able to make kits for, so that people could just like test 10 trees and here you go, and it's easy. Alternatively, you can let them grow, and in about 5 years, the blight resistant ones will get blight, or the blight resistant ones will get blight and live, and the non-blight resistant ones will get blight and die. So you'll know. So.
All right, great. Well, that is all the questions that we had. I don't know if we want to wait a couple more minutes for questions, or anyone has any last burning thoughts if you'd like to put them in the next 30 seconds. I think we can field those.
While we give folks a couple of minutes to put their thoughts and questions into the chat, if they still have any, I want to take a minute, first of all, to thank Dr. Oakes and Dr. Sandy for a fantastic talk. I learned a bunch. I had a bunch of questions answered. I hope you all out there in the audience did too. And I'd also like to take a minute in the spirit of collaboration among different arboreta to mention that if there are folks out there involved with other arboreta, that ArbNet, an interactive collaborative community of arboreta, is working with the American Chestnut Foundation to support planting different chestnut trees across different arboreta and gardens in different parts of the country in order to help support these different research projects and our understanding of the future of American chestnuts. There are currently arboreta in the Appalachia, on the East Coast, and in the Midwest. The Morton Arboretum is one, planting out American chestnut, Chinese chestnut, and restoration chestnut 1.0, a potentially blight-resistant chestnut, back cross, and monitoring those trees. They were planted starting in 2016, and so we'll be getting results on those trees in all of those different environments over the next many years to come. And I believe ArbNet is also excited about working with SUNY ESF on the darling 58 transgenic trees when they are ready for host sites. For more information on ArbNet, how to become an accredited arboretum, how to find out more about these trees, please visit arbnet.org, and I'll put all that in the chat. And if you're really excited about Arboretum, please join us for a virtual conference this fall. Registration opens June 15th, and it'll be taking place September 14th through 16th.
I think it's worth mentioning that the transgenic tree that was called Darling 58 has been named as a cultivar, and its name is Darling in single quotes. That's how you refer to a clone. So people will be able eventually to buy the cloned Darling tree, which will have one copy of the gene. And I like having trees named as cultivars.
I like that too. And then let me just say thank you to all of you for attending tonight, and issue you all a warm invitation to come and visit the different arboreta and gardens hosting this event, either in person, we hope you'll come visit us in person, or online. And all of us have different online education programs that we would love to see you at some point in the future. So I'll put those links in the chat as well. Would anyone else like to share any closing thoughts tonight while I do that?
I would just like to say thank you to our speakers tonight. The presentations were excellent, and the questions that you answered really answered a lot of the questions that I had. So it was fantastic. Thank you both.
Thank you for having us. This is great.
I'm
All right.
always happy to talk about chestnut.
All right. So without further ado, then, I think we will wrap it up for the evening. Thank you, everyone who joined us tonight. And thank you again to our speakers. And everyone have a wonderful evening. Thank you. Bye.
Hi, everybody.